Anthoceros. It is commonly called hornwort. It has horn-like or bristle-like sporophyte, so it is called as hornwort. Systematic position of Anthoceros. It belongs to the division Bryophyta. Class Anthocerotopsida, order Anthocerotales, and family Anthocerotaceae, and the genus Anthoceros. Habitat and distribution. It is amphibious in nature. It is found in moist, shaded region. It is distributed in subtropical warm temperate regions. It is not well adapted to resist dry conditions. Some common species of Anthoceros: Anthoceros himalayensis, Anthoceros erectus, Anthoceros chambensis, or grown in western Himalayan region. Anthoceros assamicus, grown in Assam. Anthoceros shvyanandani, grown in Kerala. Anthoceros also found in Mussoorie, Kulu, Manali, Chamba Valley, Punjab, Madras, and plains of South India. Vegetative and reproductive structure of Anthoceros. Vegetative structure consists of sporophyte and gametophyte. Gametophytic plant body is dorsiventrally flattened thallus. It is also looks like rosette, which appears like circular cluster of thallus. It does not have any apical notch. It is not stretched. It is smooth and it is thick in the middle and without any distinct midrib. Sporophytic plant body. The horn-like structure present on the gametophyte is called sporophyte. Anthoceros shows dehiscent sporophyte which have splitting at the tips. These horns are attached to the thallus by means of sheath-like supporting structure at the base called involucre. Involucre is a membranous envelope. There are numerous horns are present on the gametophyte. In the month of September and October, the mature thallus has erect, elongated, cylindrical sporangia. In dorsal view, we can see the greenish horn-like sporophyte and flattened gametophyte. In ventral view, we can see the rhizoids. Anthoceros have only smooth-walled rhizoids. These rhizoids helps in fixation and absorption of water and nutrients. Rhizoids are unicellular. It does not have any scales and tuberculate rhizoids. Internal structure of thallus. Thallus is not differentiated into photosynthetic or storage region. Each cells of thallus perform both photosynthesis and storage. All cells have lens-shaped chloroplast. So all cells perform photosynthesis. Number of chloroplasts varies in different species. It does not show any air chambers. The outermost layer of thallus is upper epidermis. It is continuous in nature. It shows chloroplasts which perform photosynthesis. Followed by upper epidermis, thallus shows multiple layer of compactly arranged parenchymatous cells which consist of chloroplast. Pyrenoids are also present within the chloroplast. These pyrenoids are unique feature of bryophytes called anthocerotales. Pyrenoids are microcompartments within chloroplast of algae and Anthoceros. Then schizogenous cavity. Schizogenous cavity are also called as mucilage cavity, which is present in between these parenchymatous cells of thallus. These cavities form due to breakdown of the cells. These cavities are filled with mucilage and are called mucilage cavities. These cavities open on the ventral surface through stroma, like slits or spores called slime pores. Each slime pore has two guard cells with thin walls. These guard cells do not control the size of the pore. The pore remains completely open. The slime pore represents the vestigial remnants of a previously existing aerating system. With the maturity of the thallus, the mucilage in the cavities dries out. This results in the formation of air-filled cavities. The blue-green algae nostoc invades these air cavities through slime pores and form a colony in these cavities. Thallus supply carbohydrate to nostoc. Nostoc in turn provides nitrate nutrients by fixing atmospheric nitrogen. Thus, Anthoceros shows symbiotic relationship with algae. Then, lower epidermis. It is also continuous. Some cells of lower epidermis gives rise to rhizoids. This picture shows the cross-section of thallus. Peers assume that the thalli without nostoc grow better than the ones containing the endophytic algae. But the Rodgers and Stewart said that it is a symbiotic association. But the presence of nostoc colonies in the thallus of Anthoceros is beneficial for the growth of gametophyte is not definitely known. Then the reproductive structures. Anthoceros shows vegetative mode of reproduction. They are fragmentation, tubers, gemmae, persistent growing apices, and apospore. In fragmentation, the older portion of the thallus starts disintegrate due to aging. As it reaches up to the place of dichotomy, the lobes of thallus get separated. Thus, detached lobes develop into the new plant by apical growth. This method is not so common in Anthoceros. Then by tubers. Under unfavorable condition, the marginal tissues of thallus get thickened and form perinating tubers. They may develop behind the growing points or along the margins of thallus. The tubers have outer 2 or 3 layers of corky hyaline cells which enclose the tissue containing oil globules, starch grains, and aileron granules. These structures have a capacity to pass on the unfavorable conditions. On resumption of favorable condition, tubers develop into new thalli. Then by gemmae. In some species, many multicellular stalk structures develop. These structures are called gemmae. When it detached from the parent thallus, each gemma develops into new thallus. Then by persistent growing apices. Due to prolonged dry summer, or if the plant is towards the end of growing season, the whole thallus of Anthoceros dries and gets destroyed, but except the growing point. Then this growing point grows deep into the soil and becomes thick under unfavorable condition. Then it develops into new thallus. Then by apospore. In Anthoceros, unspecialized cells of many parts of sporogonium form gametophytic thallus. This phenomenon is called apospore. Then sexual reproductive structures. Some species of Anthoceros are unisexual and some are bisexual. The male sexual reproductive structure is called antheridium and the female sexual reproductive structure is called archegonium. Formation of sex organ depends on the photoperiod. They are short-day plants. Usually, sex organ develop in winter. Then antheridium. Antheridium develop earlier than the archegonium. Antheridium embedded in the dorsal surface of thallus. Mature antheridium has a stalk and club-like body. Stalk of antheridium attaches to the base of antheridial chamber. Stalk may be slender, composed of 4 rows of cells. The single antheridial chamber may consist of many antheridia. Single-laid sterile jacket encloses the mass of androcytes. These androcytes metamorphose into anthrozoids. In some species, this jacket layer may have 4 tiers of cells. Each cells of jacket consists of plastids At maturity, these plastids change their color from green to red to bright orange. Therefore, young Antheridia are green and the mature one is bright orange or reddish. Then the anthrozoid. Mature anthrozoid is unicellular, uninucleate and biflagellated, have linear body. The flagella are of almost the same length as the body. This picture shows the dehiscence of Antheridium. Then the female reproductive structure, archegonium. It is the female reproductive structure. It develops inside the thallus. The place of the archegonium on a thallus can be identified by the presence of mucilage mound. A mature archegonium consists of 2 to 4 cover cells, an axial row of 4 to 6 canal cells, a venter canal cell and a egg. The jacket layer is not distinct from other vegetative cells like other bryophytes. Then sporophyte. A mature sporophyte consists of foot, seta and capsule. Capsule further differentiated into columella, archisporium and capsule wall. Sporogonium appears like bristle or horn, hence the species is called hornworts. Foot. It is bulbous, multicellular, consists of mass of parenchymatous cells. It acts as haustorium and absorbs food and water from the gametophytic cells for developing sporophyte. Then seta. Seta is represented by the meristematic zone. It is present at the base of capsule. It consists of meristematic cells and constantly add new cells to the capsule. The presence of meristem at the base enables the capsule to grow for a long period. It is a unique feature of Anthoceros and is not found in any other bryophyte. Then capsule. Capsule is further differentiated into columella, archisporium and capsule wall. The columella region is a central sterile pan. It extends nearly to its tip. It is made up of 16 vertical rows of cells. In transverse section, it appears as a solid square. It provides mechanical support, functions as water conducting tissue. Then archisporium. It is present between the capsule wall and the columella. It extends from base to the top of the capsule. In upper part of the capsule, archisporium differentiated into sporogenous tissue which produces spores and pseudoelaters. These pseudoelaters are unicellular or multicellular and branched or unbranched. Then capsule wall. Capsule wall consists of 4 to 6 layers of cells, forms the outermost layer of epidermis. It has cutin deposition on their walls. The epidermis is discontinuous because of presence of stomata. Then each stoma consists of pore and 2 guard cells. The cells of inner layer have intercellular spaces and contain chloroplast. The sporogonium is partially self-sufficient to synthesize its own organic food, but partially it depends on the gametophyte for food. Then spores. Columella is the mass of spores and pseudoelaters. The 2 walls that separated and twist hygroscopically, elaters become dried and loosen the spores, thus it facilitates the dispersal of spores. Spores are haploid, uninucleate tetrad. Each spore is surrounded by 2 wall layers. The outermost layer is called exospore, which is thick and ornamented, and its color varies from dark brown to black or yellowish. The inner layer is called endospore, which is thin. Anthoceros shows resemblance with green algae because it is simple green gametophytic thallus. It have chloroplast and it has pyrenoids. Then it also shows a resemblance with liverworts because thallus is simple green gametophytic without any differentiation, and it shows similar apical growth and biflagellated anthrozoids. It also shows some advanced characters by presence of highly differentiated ventilated system in the capsule wall, and presence of columella, and have sunken sex organs.